Today we're going to explore mitochondrial DNA, commonly called mtDNA, and how it is used in genetic genealogy, anthropology, and deep ancestry studies. We'll discuss what mtDNA is, how it is inherited, mutations and genetic distance, haplogroups, testing methods, and how mtDNA can help genealogists trace maternal ancestry over thousands of years. Most DNA people think about is nuclear DNA, which sits inside the nucleus of the cell. Mitochondrial DNA is different. It exists inside tiny structures called mitochondria, which help produce energy for the cell. Unlike autosomal DNA, mother to child, to grandchild, both sons and daughters inherit mtDNA from their mother, but only daughters pass it forward to future generations. mtDNA follows an unbroken maternal chain. Your mtDNA came from your mother, her mother before her, and so on. This makes mtDNA incredibly valuable for studying direct maternal ancestry far back in time. Because mtDNA changes slowly, researchers can track maternal populations over thousands of years. Each DNA test serves a different purpose. Autosomal DNA helps identify recent cousin relationships across all family lines. Y-DNA tracks direct paternal ancestry and is only carried by biological males. mtDNA tracks direct maternal ancestry and can reach extremely deep into human history. Together, these tests create a much fuller genealogical picture. One of the most misunderstood aspects of mtDNA testing involves mutations and genetic distance. mtDNA mutates very slowly compared to many other forms of DNA. On average, a single mtDNA mutation may occur approximately once every 1 to 30 generations. That is a huge range. This means two people with an exact mtDNA match may still share a common maternal ancestor many hundreds of years ago. Likewise, people separated by only 1 or 2 mutations could potentially still connect within genealogical timeframes. Mutation rates are estimates, not exact clocks. Genetic distance measures how many mutations differ between two mtDNA tests. A GD0 match means no detected mutations separate the two testers at the tested level. However, because mtDNA mutates slowly, even a perfect match could trace back hundreds of years, or potentially much farther. This is why mtDNA should always be combined with traditional genealogy and historical documentation. As mutations accumulated over thousands of years, major maternal branches formed. These branches are called haplogroups. Each haplogroup represents ancient population migrations and maternal line expansions across the world. Some haplogroups are commonly associated with Europe, Africa, Asia, or indigenous populations of the Americas. However, haplogroups alone do not prove tribal affiliation or ethnicity. Context and documentation still matter enormously. Earlier mtDNA tests focused only on small portions of the mitochondrial genome. Modern full-sequence testing examines the entire mitochondrial DNA molecule. This provides more accurate matching, deeper haplogroup assignment, better mutation analysis, and improved genealogical usefulness. For serious maternal line research, full-sequence testing is generally preferred. mtDNA is especially useful when researching unknown maternal lines, adopted ancestors, female line brick walls, colonial populations, and deep ancestry projects. Because surnames often change through marriage, maternal genealogy can become difficult to track through records alone. mtDNA provides another important layer of evidence. mtDNA is powerful, but it also has limitations. Because mtDNA follows only one direct maternal line, it represents a tiny fraction of your total ancestry. You may have thousands of ancestors in a time frame where mtDNA reflects only one maternal path. This is why responsible genealogy combines DNA evidence, historical records, geography, social history, and critical analysis. One of the most widely used platforms for mtDNA testing in genetic genealogy is FamilyTreeDNA. FamilyTreeDNA offers full mitochondrial sequence testing, maternal match databases, migration tools, and advanced haplogroup reporting. Many serious mtDNA researchers use this platform for maternal lineage studies. mtDNA gives us an incredible glimpse into humanity's maternal past. It connects modern people to ancient migrations, deep ancestry, and maternal lines stretching back thousands of years. But, like all genealogy tools, it must be used carefully and responsibly. DNA is strongest when paired with documentary evidence, historical context, and thoughtful analysis. Thank you so much for joining me for this discussion on mitochondrial DNA and maternal ancestry research. In future videos, we'll continue exploring chromosome mapping, autosomal DNA, Y-DNA studies, haplogroups, Native American genealogy, historical records, and responsible genetic genealogy research.